Welcome to our biology lessons with Dr. Hanaa. Today we will solve with each other November 2025 variant 2 paper 6. Question 1. A student investigated the rate of respiration in yeast using different concentrations of glucose. They used a 5% glucose solution to make 1% glucose solution and 4% glucose solution. So, from the first sentence, who can tell me what is the independent variable? We know that the independent variable or the changed variable. So, we will use different concentrations of glucose. So, this is independent variable or changed variable. We want to know its effect on the rate of respiration. Okay? The student used this method, label 2 small test tubes 1% and 4%. Use a syringe to put 1 cubic centimeter of 5% of glucose solution into small test tube labeled 1% and put 4 cubic centimeter of 5% of glucose solution into small test tube labeled 4%. Use a clean syringe. So why he use here a clean syringe, not the same one? To prevent the contamination or can change the concentration that is needed. So use a clean syringe to put 4 cubic centimeter of distilled water into small test tubes labeled 1 and 1 cubic centimeter of a distilled water to the small test tube labeled 4. Pour yeast suspension into small test tube labeled 1% until it completely full. Keep the small test tube upright and carefully place a large test tube over the top of the small test tube labeled 1% and push the small test tube up like the figure. Again, so in front of us, the small test tube containing yeast suspension and glucose solution and put it in an opposite way with a large test tube, in an opposite way. And just press with your finger to hold the small test tube in its position. And then you have to invert this test tube to be like it exists in B. And then we will put it in water bath containing warm water. Put the large test tube and small test tube labeled 1% into the warm water bath. So we have a source of error here. They didn't mention the value of the temperature of the water bath, and he didn't maintain this temperature. So how can we maintain the temperature? By using thermostatically controlled water bath. Start the stop clock and wait for 5 minutes. This is controlled variable. After 5 minutes, stop the stop clock and use the ruler to measure the height of the yeast suspension in the large test tube. This is the height that's needed to be measured. Remove the test tube from the warm water bath and discard them. Pour the yeast suspension into small test tube and label 4% until it's completely full. The same steps that we did it before for 1%, we will make it for 4%. Figure 1.3 is a diagram of the water bath in step 8 and 11. So one of them for the glucose 1% and the other for the 4% of glucose solution. So we have 2 different concentrations. Measure the height of the yeast suspension in each of the large test tubes. So we will measure this height of 1% of glucose solution and this height for 4% of glucose solution. So measure the height of the yeast suspension in each of the large test tubes shown in the figure. Prepare a table for the result and record your measurement. So what are the data that we needed to make or to prepare the table? So we have two different concentrations, so 1% and 4%, and also the height of yeast suspension by millimeter. We have 1% and 4%, and just measure the height of yeast suspension using millimeters. Sometimes your measurement is not exactly the same values that can find it in the mark scheme because the printed copy is totally different from the paper exam. So no problem if you find a difference between them, your measurement is correct. State the conclusion for the result. So we have to know that when we start to write the conclusion, we have to return back to the objective of the experiment. So we have to know that the different concentration of glucose solution, so this is the independent variable, you have to know its effect on the rate of respiration. We return back to the original objective, this one. So when we start to write the conclusion, we have to refer to the concentration of glucose by increasing or by decreasing, so whatever, and its effect on the rate of respiration. So how can we write it? The higher the concentration of glucose, the higher the concentration of glucose solution, the greater the rate of respiration in yeast. Next question, state the independent and dependent variable in this investigation. We get the independent variable from the first sentence. So about this is the concentration of glucose. So we have different concentrations of glucose. And we just measure the height of the yeast suspension. So this is dependent variable to know that the rate of respiration. So the dependent variable, the height of yeast suspension. Describe how the temperature of the water bath could be maintained. That we said it when we explained this investigation. So we said it by using thermostatically controlled water bath. Next question, he asked about why a clean syringe was used in step 3. We said that if we use the same syringe that we put it, the glucose volume, and the same one we put the distilled water, so we will change the concentration. So it's called contamination. So we use a clean syringe to avoid or to prevent the contamination. A student extended the investigation by using more concentration of glucose solution. Calculate the volumes of 5% of glucose solution and distilled water that are needed to make 5 cubic centimeter of 3% of glucose solution. So we know that the 5%, if we have the original concentration of the glucose solution, I just get it from it 5 cubic centimeter, it's concentration of 5%. So how much cubic centimeter needed to make it 3%? So we will make it a cross multiplication. So 3 multiply 5 divided 5, which is equal 3 cubic centimeter of glucose solution. And we have to then add it to be 5. So we will add 2 cubic centimeter of distilled water. Sports drink often contain glucose, which is a reducing sugar. State how you could test the sample of sport drink for reducing sugar. So we have the Benedict's reagent or Benedict solution, and then apply heat using water bath at 80 degrees Celsius. If the blue color of Benedict solution changed from blue to green or yellow or orange or brick red, so this solution contains reducing sugar. Why we have all of these colors? Because this investigation or this test used to test the concentration also of the reducing sugar. So the most concentrated glucose or reducing sugar, so it will give us brick red, and the least one give us green. Ethanol emulsion test can be used to show that a sample of food contains fat. State the result of a positive test. So the positive test of emulsion, when we test the presence of oil or fat or lipids, so it will give us white emulsion or cloudy emulsion. Suggest one safety precaution that should be followed when using ethanol. We know that ethanol is flammable and volatile. So we have to keep ethanol away from flame because ethanol is flammable, and we have to cover ethanol container to prevent evaporation, and also you have to wear goggles. So we have the priority first for the flammable, so we have to keep it away from the flame. If you just added more points, so you have to cover it or wear goggles. Next, the question to plan an investigation to determine the effect of exercise intensity on the heart rate in humans. In this investigation, you have to know that the exercise intensity is independent variable, so we can change it by ask the people for walking, for running, for jogging, so different intensities of exercise. On the heart rate, how can we measure the heart rate? How can we convert the heart rate into dependent variable? So we can using heart monitor or ECG, or counting the heartbeats at a certain time. So in front of you, many options to write it as a dependent variable. Firstly, this exercise or this investigation for people. So when we ask people to do an exercise or to do the activity, so they should have the same age, same mass, same gender, and fitness level or activity level. And then we have to measure their heart rate at rest at the beginning of this investigation using an ECG. What is ECG? This is called electrocardiogram, that's used to monitor the heart rate, or also can count the number of heartbeats in a certain time. And then ask them to walk for 5 minutes with the same intensity of walking and same environmental temperature to not affect on their heart rate. After that, we have to measure their heart rate directly after walking using, again, ECG. After doing this walking, ask them to rest for 5 minutes or 10 minutes to return back to their normal heart rate. After that, we have to repeat all of these steps above, once for running and other for jogging. Okay? Repeat this activities or this investigation 3 times for each exercise intensity and take an average. And people should be well hydrated, wearing comfortable shoes, comfortable sportswear, and not suffering from any health issues. Next, the question, the figure 3.1 is a photograph of a necklace starfish. Okay? He asks you to draw a large diagram of a necklace starfish like this. So please, you have to put in your mind the main important points and analysis for this diagram. So how many arms for this starfish? Okay, and you can do it by this way. We have an arm here, and then another one, the third, fourth, fifth. In one straight line. If you have a problem with the drawing, so no problem to erase it and redraw it again, but don't press on your pencil. It is preferable to use a pencil with the HB. This is the degree of graphite. Okay, not more dark or not lighter. So it is in the middle. And then if you concentrate well, we can find them 5 arms. And some of the arms is dark and the other is light. So we can make any shaded area to be separated from each other. And then we have a central disk in the middle. Just like that. So if you find this any problem with the drawing, and I think this is usually a mistake, you can erase it and make it where there is no intersection. So when we start to draw like this, no, it is not preferable to do it like this. So what we're going to do, like this, and be careful and be slowly to join them together. And this is my drawing. Line PQ in figure 3.1 represents the diameter of the necklace starfish. Measure the length of PQ in the figure. So you have to bring your ruler and measure from P to Q. I know that you use your ruler in centimeter, but you have to convert it into millimeter. And also you can find your answer is totally different from the value in the mark scheme, so no problem because the printed copy is totally different from the paper exam. So your measurement is correct, but you have to convert it into millimeter. So my answer is 93 millimeters, so calculate the actual diameter of the necklace starfish. They give us the rule, but I like my magic triangle that said I am. This is magic triangle for the magnification. So I represent the image, A represent this actual length, and LM represent magnification. So just circle that, the part that you needed, and this one means divide, and in between it means multiply. So when we need an actual length, so image divided magnification, from where we can get the magnification? So we can find it 0.32. So 93 over 0.32. So it will give us 290.625. And he wants this two significant figure. So 290. Figure 3.2 is a photograph of spiny starfish and necklace starfish. State 2 spiny. State 2 ways the spiny starfish differ from the necklace starfish, and one way is similar. So they ask us to write the similarities and differences, just two differences and one similarity. So we can find here this rough surface or spiny surface on this one, but this one is smooth. So spiny starfish has spines or rough surface, while the necklace starfish is smooth or does not have spines. So we can find the central disk or the central part here is lighter than necklace starfish. Okay, the book can tell me similarities between them. Both have 5 arms, so 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. So both have 5 arms. And they have circle in the middle. So whatever this is light or dark, the circle in the middle. Okay, so whatever this is the point of differences, this is dark and light. And the similarities both have this circle. This is called the central disk. And they have radial symmetry. So what's a radial symmetry? This radial, it means that it looks like the radius. So we have this is equal to each other. This is radial symmetry. Both. Okay, from the center, 1, 2, 3, 4, 5. So they have radial symmetry also. Saved armed starfish live in the sea. Scientists investigated the distribution of 7 armed starfish in one region. So this is not very. Photograph of 20 meter square area of seabed were taken at different depths. So this is the same area, same size of area or same area, which is 20 meters square. The depth in the water in each area was measured. When the water at its highest, okay, the number of 7-armed starfish in each photograph were counted. All photographs were taken on the same day in July. Also, this is controlled variable. So we have controlled variable, controlled variable, controlled variable. State two ways that the method used by the scientists ensured that the number of 7-armed starfish counted at different depth. Different depth, this is independent one, could be compared because we just have only one different, but all the other factors exactly the same. So when we have an experiment, we have to change only one variable to know its effect. Not more than one variable should be changed because we never know which one of them that affect the result. It just we have different depth in the same region, at the same area, at the same highest level of water or at high tide and the same day in July. So just only one factor that change it, which is different depth. So the answer, as we just said now, the same area, the same size of area, which is 20 meters square, and same day, the same tide of, same time of tide. The results of the investigation are shown in the table. They give us the depth by meter, and we can find that if you concentrate well, we can find it, it's in a range form. It is in the range, it's a range. We know that if we find any range, so we have to draw histogram or frequency diagram. What is a histogram or frequency diagram? This is diagram, we can use it in a bars touch each other. There is no gaps between them, so it's a range by using the depth. And so we will put the depth on the x-axis. Okay, because this is independent variable, something that only change it here, which is independent variable, and we have to put it on the x-axis. And then we have to count the number of 7-armed starfish in this area. So the question is, plot a histogram. I just said the histogram, exactly the same idea if they ask about the frequency diagram, exactly the same on the grid on the data. So I draw it by this way. Depth. Okay, the depth. We'll just try to make it large as much as possible. So we have the depth in millimeter and the number of 7-armed starfish counted. And then we have to put below each column its identification or its range from 0 to 9.9. Next column from 10 to 19.9. Next column, because each data below it, this was labeling for this column. Okay, so the first column, it's the range from 0 to 9.9. The second column from 10 to 19. So this is give us, it looks like a label. Okay, don't make it this arrow, just show you to just understand it, but don't draw this arrow. And then we have to make the column. This column should be equal in the width. So the width this width should be equal to each other. This all of them. No one is greater than the other, should be equal. All of them should be equal to each other. And there is no gaps between the bars because this type of histogram or frequency diagrams, so including a range, so we have here a range. So we have to draw it by this way. State the depth range in table 3.1 at which most 7 armed starfish were found. So which one of them is the highest? Which one of them? Yes, from 10 to 19.9. This is the highest value. So we have to write it also in the form of range. Using the information in the table 3.1, calculate the total number of 7 armed starfish, the total number, that were photographed at all depths. Use this value to calculate the percentage of starfish that were photographed in this area. So at the beginning, we have to calculate the total. So total sum of all starfish that exist in an area, and we have to know the percentage of the range from 0 to 9.9. So firstly, we have to make the total, this total sum of all of them, which is equal 3,428. And then how can we get the percentage? So the value of from 0 to 9.9, where is it? 337 divided the total multiply 100, which is the percentage of the starfish in this area. It gives us this 9.83 and just becomes the nearest whole number, which is equal 10. Now we reach to the last part of the exam. I hope that you enjoy it and waiting for more and more exam. Thank you and goodbye. Don't forget to like, share, and subscribe to find all the new exams. Thank you and goodbye.